Okay, so I'm Andrew Vigotsky from Northwestern University. First, I'd like to apologize. My slides are very impersonal, but this is science, so it's fine. Okay, so I study pain psychophysics, specifically a quirky psychophysical phenomenon. So with typical psychophysics, we think about just a classic dose-response relationship between a stimulus intensity and the perception. So if I apply a low temperature of heat to your forearm, you might not feel any pain. Then as I increase the temperature of the heat, you'll feel more and more pain. And there's a simple dose-response relationship. But in engineering, we would call this quasi-static. So it assumes that the system is static and that things aren't changing in time. But in reality, things are a bit more complicated. So in reality, if you do this in time, there's this dynamical phenomenon where if you take into account the history dependence, things aren't straightforward. And so this phenomenon that you're seeing here is called offset analgesia. And basically what it is, so you can see the temperature in red. So if I apply a moderate temperature, you'll feel some pain. If I increase the temperature a little bit, you'll feel more pain. And then if I decrease the temperature back to what it was, you'll feel no pain. So there's this complete analgesic response. And that's called offset analgesia. So it's the disproportionate decrease in pain for a small decrease in the intensity of the noxious stimulus. And interestingly, this is related to the clinic. So if you try this in patients with chronic pain, they don't experience as much of an analgesic effect. So it's blunted by about 30% or so. And we really don't know why this occurs. We don't know the mechanisms. So the topic of my dissertation is what causes this disproportionate decrease in pain or offset analgesia. So previous literature suggests that central mechanisms are sufficient for this. I'm not going to go over that literature, but there are two hypotheses, so namely the opioidergic and the NMDAergic hypotheses that were kind of mainstream for a bit. So the opioidergic hypothesis is that descending inhibitory systems that are controlled by opioids control this effect. And so pharmacological studies have shown if you give someone an opioid agonist or antagonist, it doesn't seem to modulate the dynamics or the magnitude of the effect. So opioids are out. NMDA is associated with something called diffuse noxious inhibitory control in the brainstem. And if you give people ketamine, which is a strong NMDA antagonist, it also doesn't change the effect. So it seems like it's not NMDAergic either. So what I'm proposing is that it's dopaminergic, and I'll explain why. So dopamine it's an interesting system, so there's a lot going on. But if you look at VTA dopamine neurons and you apply a noxious stimulus, when you apply the noxious stimulus, there's a decrease in firing. But interestingly, when you stop the stimulus, there's an increase in firing of VTA dopamine neurons. And that corresponds with an increase in dopamine release in nucleus accumbens following the noxious stimulus. And you see something similar in humans in fMRI. So when you decrease the intensity of a noxious stimulus, or when you remove the noxious stimulus rather, you see activation of nucleus accumbens. So that's kind of what this is showing and what's interesting here. So this is the decrease in the noxious stimulus, so the stimulus turns off. And in healthy individuals, you see activation of nucleus accumbens. But in people with chronic pain, you see deactivation. So to me, this says, okay, maybe there's something here, and maybe it could link offset analgesia to the mechanisms of chronic pain. And this was a study that was done in 2018. It's not the greatest since their effects weren't super strong of offset analgesia, but what you see here is kind of similar findings to what I just showed. So chronic pain patients have decreased activation during the offset analgesia period. Healthy controls have increased activation. So all of that seems consistent. But this is kind of weird because, like, how exactly would this happen? We don't really think of dopamine as a system that modulates nociception. So first we have to understand what's going on in the dopamine system. And in 2009, this paper came out, and there have been a ton of papers since then. But dopamine is not this monolithic neurotransmitter that's just associated with reward and motivation. It's a lot more complicated. There are different dopamine circuits. So if you provide a foot shock to a mouse, there are some dopamine neurons that are inhibited, and there are other dopamine neurons that are excited. So there are these aversive responding dopamine neurons that are implicated in pain, presumably. And Stefan Lammel's work shows this really nicely in that there are certain hotspots within the mesolimbic system that seem to be more specific to aversion. And there's other work that shows that there are hotspots that are more associated with reward. So these are kind of complementary systems within the mesolimbic dopamine system. So what I'm proposing is that when a noxious stimulus comes in, it gets transduced to nociception, and that nociception projects to the aversive dopamine neurons. So this has been shown by Lammel's group and others. And not only that, but the change in nociception also serves as input to those aversive dopamine neurons. And I also assume that aversion is necessary for pain. So in addition to that, what I'm proposing is that the negative derivative of nociception feeds into the rewarding dopamine neurons. And then I'm suggesting that they interact with each other as well. So if reward inhibits aversion, then when there's a decrease in nociception, reward activates, that inhibits aversion. And if aversion is inhibited, then pain should shut off. So if these two systems interact, then it could explain offset analgesia. So I created this model, I derived the differential equations that are associated with it, and what's nice is this forms as a nice mathematical hypothesis for what could be going on. And this is a simulation, or an integration of the results. So if I apply a noxious stimulus, you get aversion activation that's in red. Then when the stimulus turns off, you get the blue, and that shuts off the aversion. And then you can see that this creates an offset analgesia response. And this is what it looks like fit to real data. So black is real data, and green is self-report. So it fits the real data pretty nicely. So it seems to explain this phenomenon from a behavioral perspective, but we need to link it to the nervous system. So that was the purpose of this study. So I collected data on 14 young, healthy individuals. So what I did was I applied the offset analgesia stimulus to their forearm while they were in the scanner, and they were not rating the stimulus, because arguably rating the stimulus is a different task, and if they're doing a different task, that can confound your measures. So then I applied the stimuli again when they were outside the scanner, and I obtained ratings. And then I fit the data with my model, and I modeled each voxel nucleus accumbens using my aversion time series and my rewarding time series. Importantly, these data that I'm going to present are pretty preliminary. I haven't fit this model to each person. It's pretty difficult to do so, and I'm still working on that. And I also have a larger sample that I'm working with now. So these are the results. So each color represents a t statistic. So negative means that it's negatively associated with the reward or aversion, or this is the difference between the two. And then positive is the bright yellow. But something that's clear is that it's not a homogeneous activation across nucleus accumbens. And what's interesting is that there does seem to be some structure here, especially in the difference between the two. Although this isn't quite the structure that we expect, but I'm not, so specifically I would have expected this to be reversed. But I still need to perform more analyses and do this properly, modeling each subject's time series specifically. But what's important is we are kind of recapitulating the concepts in the literature in that there is some regional dependence of this effect. And I'll have more participants with more balance, so 9 females instead of 4 females, with subject-specific parameters. And I'm also running a crossover trial with methylphenidate, so that's a dopamine reuptake inhibitor. So if there's more dopamine available in the synapse, that raises the question, does it change the dynamics of this effect? So I'm doing that with a placebo control. So thank you. So it's I guess because I don't have any names on here because it's impersonal, to the Aptarian Lab and also my funders, National Science Foundation, National Institutes of Health. Thanks.